Chapter 13, Modern Understandings of Inheritance. Here is a karyotype, a layout of a person's chromosomes. This is considered a normal biological male as all chromosomes are present and the sex chromosome is XY. We now know that genes express our traits. During Mendel's time, he didn't call these genes, he called these factors. And about 50 years later, chromosomes were discovered to be the objects that carried genes. In this chapter, we will see how the effects of chromosomal inheritance can actually affect an offspring's appearance. After Mendel, microscopy improved to the point that biologists were able to see the stages of mitosis and meiosis in cells. They visualized the uncondensed amorphous chromatin develop into an X-shaped chromosome. It was almost 50 years later before Mendel's discoveries involving P genetics were reevaluated and linked to chromosomes. In 1902, Theodore Boveri, on the right, observed in sea urchins that embryonic development does not occur unless chromosomes are present. Later, Walter Sutton, on the left, observed that chromosome separation in the daughter cells happened during meiosis. Together, these two developed our understanding on the chromosomal theory of inheritance. The following observations about chromosomal theory of inheritance was developed by Boveri and Sutton, and they are: during meiosis, homologous chromosome pairs migrate to discrete structures that are independent of other chromosomes. Second, chromosomes sorting from each homologous pair into pregametes appear to be random, hence crossing over and independent assortment seen in prophase 1 and metaphase 1. Each parent synthesizes gametes that contain only half their chromosomal complement. That is, parents make haploid sex cells. Even though male and female gametes differ in size and morphology, they have the same number of chromosomes, suggesting equal genetic contribution from each parent. And finally, the gametic chromosomes combine during fertilization to produce offspring with the same chromosome number as their parents. Combining haploid cells provide a diploid zygote with the same chromosome number as parents' somatic cells. Chromosomal theory was already developing after Mendel's experiments, but the observations of Boveri and Sutton helped provide concrete evidence for this. Thomas Hunt Morgan, as I mentioned in the previous chapter, worked with fruit flies, and he was the one to provide the first experimental evidence for the chromosomal theory of inheritance. So while Mendel was able to show that all of his traits were inherited independently of each other, he didn't know that his 7 P traits were held on separate chromosomes. Our understanding of chromosomes show that genes can be linked together if they fall on the same chromosome and are close to each other on that same chromosome. Franz Johnson was the first to observe the change of chromosomal parts during crossing over. He also saw that genes can become unlinked during its time and recombine on the homologous chromosome. So let's say you inherit a homozygous dominant set of paternal alleles and a homozygous recessive set on the maternal alleles for traits A, B, and C. This would actually make you heterozygous for all 3 traits. We know that crossing over can and does occur during meiosis 1, so here are the different combinations of chromosomes that potential sperm or egg cells can have based on these genes in one individual. If Mendel was correct and all genes were transmitted independently with one another, we would expect the same ratios of the 8 types of chromosomes displayed above. However, these types are more likely to show up in gametes pointed out by the yellow arrows. For one, crossing over does not always occur. And the second one, B and C are often inherited together because they are closer to each other on the chromosome than they are to part A. And that makes it higher likely that the gamete will receive both alleles from the same chromosome when crossing over occurs. So we call parental types any kind of gamete with chromosomes that match the original chromosomes that you have, and in non-parental types are what we call recombinants. So recombinants are just a mix and match of the 2 chromosomes of the homologous pair. Thomas Hunt Morgan tested chromosomal theory on fruit flies concerning 2 genes. He called offspring that resembled either parental offspring, and offspring that created a new set of genotypes recombinant offspring. The following diagram displays the three hypotheses developed by Thomas Hunt Morgan concerning two genes tested on fruit flies on their relationship to chromosomes, followed by the actual results. So in the first part, part A, the genes are found on different chromosomes. Therefore, the offspring have a 50% chance of having recombinant or inheriting a different combination of traits than the parents, or a 50% chance of inheriting the same combination of their parents, also known as the parental types. But this would assume that these genes are found on separate chromosomes. In part B, both genes are found on the same chromosome, but no crossing over occurs in meiosis. Therefore, 100% of the offspring will have the parental types and no recombination will exist. In part C, both genes are found on the same chromosome and crossing over occurs in meiosis. Therefore, 50% of the offspring will have the parent type and 50% will have the recombinant type. But however, part D shows that Morgan's actual results from his experiments with fruit flies concerning these genes were not 50-50. He thought that crossing over would occur, it would cause a 50-50 split between the recombinant versus parental offspring, but he found out that technically the majority of the offspring were the parental genotypes and a very small minority ended up being recombinants. So why is that? So why does crossing over only occur sometimes and not others? Well, it depends on the distance between the genes on the same chromosome. So the further two genes are from each other on a chromosome, the likelihood recombination occurs. It just so happens that in Morgan's fly study, the genes being studied were really close to each other on the same chromosome, significantly reducing recombination in offspring. In fact, this is quantified by a recombination frequency. The higher the number, the higher the likelihood of recombination between two alleles on the same chromosome. So we can picture this using what's called a simple little genetic map. And this shows the locations in map units called centimorgans, or C-M for short. So 1 centimorgan is equivalent to a recombination frequency of 0.01 or 1%. In other words, the chances that those two genes will be recombined. Take the likelihood that wing type and eye color are recombined. All you have to do is subtract 65.5 minus 57.5 and you get an 8 centimorgan, or an 8% chance of recombination. Now compare this to the recombination frequency of wing type and body type, and you end up getting a 17 centimorgan difference, or 17% chance. So your chances of recombination increase the further the genes are away from each other, and the chances of them not recombining are less, because them being closer together makes it easier for them to be linked together whenever crossing over occurs. I can compare the recombination frequency of wing type and body color using the following formula. With Morgan's flies, I take the recombinant types and divide by the total offspring and get a recombination frequency of 0.169, or 17%, which is close to the difference in centimorgans on the chromosome map. There is a correlation between map unit distance and recombination frequencies. However, this correlation is limited to a recombination frequency value of 0.5 or 50%. Beyond that, the frequencies become less accurate. That's because greater distances means more unpredictable crossovers. And what I mean by that is if you were to take the eye color here in Aristay, which is 104.5 minus 0, that's 104.5 centimorgans, which ends up being a 104.5% chance, which isn't really possible mathematically. And so anytime you go above 50% or 0.5 frequency, you're going to end up with some more unpredictable crossovers. So it only works when it's less than 0.5 or 50%. So what that means is because eye color and wing type are closer to each other, their chance of being affected by crossing over is less than wing type and body color. This means that wing type and eye color alleles will more likely be inherited together as they're more linked than wing type and body color. We can conclude that the reason why all of Mendel's pea plant traits successfully assorted independently is that all traits are found on separate chromosomes, or if they're on the same chromosome, They are too far apart to successfully link together. Up next, we're going to be looking at some chromosomal basis of inherited disorders.